Hi, and welcome to my presentation on Chapter 19, Blood. So first we'll go over the functions of blood, then we'll get into the components. Blood has a lot of functions. Of course, it functions in gas exchange, carrying oxygen and carbon dioxide around the body, and allowing those gases to be exchanged with the tissues. It also transports other solutes and distributes them around the body. That would include all of your nutrients and vitamins, most prominently glucose, but also other things like amino acids and lipids, and of course the vitamins. It also distributes hormones around the body, waste products, which would include CO2, but also other things such as urea would be transported in the blood to the places where they will be eliminated from the body. And then finally, ions like sodium and potassium are carried in the blood. The blood also functions in immunity. A lot of the cells in the blood are white blood cells that have immune functions and are part of the immune system. You also have proteins in the blood that are part of the immune system. And so the blood is carrying these cells and proteins around the body and allowing them to access all the different tissues of the body whenever they need to. The blood also functions in maintaining body temperature. So blood carries heat, and depending on whether it's hot or cold outside, the body can vary how much blood is running through your skin, and that will affect how much heat you're losing from the blood to the environment, helping you to maintain your body temperature. The blood functions in hemostasis, which is the sciencey name for blood clotting. So anytime that you have an injury to the blood vessels that causes bleeding, the response to that is to form a blood clot, which is the process of hemostasis that allows you to maintain the blood inside your body and to maintain your blood pressure. The blood also functions in maintaining your acid-base balance and making sure that your pH is always at the correct level. So some of the components of the blood are buffers that do not allow the pH to change very much in the blood. And then finally, you have blood pressure homeostasis. So the blood has various ways that it affects blood pressure, but most prominently the blood volume. If you have a lot more blood, then you're going to have a higher blood pressure. If you're losing blood volume, then that's going to reduce blood pressure. Then of course hemostasis is also a necessary response to maintain your blood pressure. If you have an injury to a vessel and you're bleeding, that means you're losing blood. That's going to drop your blood pressure, and hemostasis prevents that from happening. So now we'll go over the different components of the blood. Overall, you have about 5 liters of blood in your body, although that number varies depending on how tall you are and what your gender is. Basically, the bigger your body is, the more blood you're going to have. Blood can be divided into the plasma and the formed elements. Plasma is the extracellular matrix of the blood, so it's all the liquid components of the blood and the stuff that's dissolved in there in that liquid. Then the formed elements are the cells within the blood. The plasma is referred to as a colloid because some of the things that are in there are dissolved in the water, but some of them are not. Some of them are just suspended, and that's forming a colloid. So about 55% of the total blood volume is plasma, that liquid part, and then the other 45% is the formed elements or the cells. Most of those cells are the erythrocytes, which are red blood cells, abbreviated RBCs. Then you also have white blood cells, which are abbreviated WBCs, and those are also called leukocytes. Then you have platelets, which are also called thrombocytes. So the red blood cells are accounting for about 44% of total blood volume, which leaves about 1% left for the white blood cells and the platelets. The red blood cells are functioning in distributing oxygen and carbon dioxide. White blood cells are immune system cells, and platelets function in hemostasis. We can measure the amount of red blood cells that you have in the blood using the hematocrit. The hematocrit is the percentage of blood volume that is accounted for by red blood cells. Whenever you're going to do that measurement, you need a blood sample, then you centrifuge it, basically spinning the sample at a very high speed, and that makes the components of the blood separate based on their density. Everything that's heavy is going to end up at the bottom of your tube. Everything that's light is going to end up closer to the top. So here's your blood sample, and after it's been centrifuged, it would look more like this. The heaviest stuff in there is the red blood cells, so those all end up at the bottom. Then at the top you have the lightest stuff, which is the plasma and all the stuff, the liquid and all the stuff that's in there. That's dissolved or suspended in there. So that all goes to the top. And then in the middle, between the plasma and the red blood cells, you have what's called the buffy coat that contains white blood cells and platelets. Those are heavier than the plasma, but not as heavy as the red blood cells. So then you can just measure what percentage of this tube is accounted for by the red blood cells at the bottom, and it should be around 44%, varying some depending on your age and your gender, but about 44%. So going into more detail with the plasma, most of plasma is just water. About 90% of plasma is water. The remaining 10% would be molecules that are either dissolved or suspended in that water. Most of those molecules are proteins. So about 9% of the plasma consists of plasma proteins. The most significant protein in the blood is albumin. It's very important, and it's also the most abundant protein in the blood. The job of albumin is to maintain osmotic pressure of the blood. So it's basically making sure that you have a similar amount of solutes in the blood as you have in the tissues around the blood and in the cells that are in the blood. If you don't have similar solute levels inside the blood cells and in the blood, or inside the blood versus inside the other tissues of the body, what's going to happen is that water is going to start moving in or out of the blood into those other compartments or from those other compartments. So if you don't have enough albumin in the blood, you're not going to have enough solutes in the blood. You're going to have more solutes in the tissues, more solutes in the cells, and water is always going to follow the solutes. So it will then leave the blood and enter the cells in your blood, which could destroy them, or it would enter your tissues around the blood, which would drain the blood of its volume and lower your blood pressure. So for that reason, it's very important to have the correct amount of albumin in the blood. You also have immune proteins, which are broadly called gamma globulins. A lot of proteins in the blood are called globulins. Actually, basically it just means that they're shaped like globs. And gamma globulins would be immune proteins. You have two main types of immune proteins in the blood. You have antibodies and you have complement proteins. You guys have probably heard of antibodies before. Complement proteins, probably not. We're going to cover both of those types of proteins in more detail when we do the immune system chapter. But for now, they're just immune proteins that are in the blood. They come from white blood cells or leukocytes. Then you also have transport proteins in the blood. The three main types you have are alpha globulins, beta globulins, and lipoproteins. The job of transport proteins is to transport stuff in the blood. Anything that does not dissolve in the blood but still needs to go through the blood needs to have a transport protein that will bind it and take it through the blood. Anything that is fat soluble will not be soluble in the blood because the blood is mostly, the plasma is mostly water. So the solvent in the blood is water. So if you can't dissolve in water, you can't dissolve in the blood. So you have like fats that need to go around the blood and provide fuel to the body. So you have to have a transport protein to take those things around. That would be a lipoprotein in general. And you also have steroid hormones. A steroid is a type of lipid, so it does not dissolve in water. Your sex hormones and your stress hormones are all steroid hormones. So they don't dissolve in water, but they still have to go through the blood to the whole body. So you have transport proteins that bind them and take them through the blood to the body tissues. And then finally, you have clotting proteins in the blood. You have a lot of different clotting proteins. We're going to go over a lot of them when we go through the clotting cascade later in this chapter. One of the main ones that you have is fibrinogen. That's kind of the final clotting protein that will actually form the blood clot. So those are the proteins that you have in the blood. Those are mostly coming from the liver, except for the immune proteins that are coming from leukocytes. The rest of them would be made largely by the liver. In addition to those proteins, you have small solutes in the plasma. Only about 1% of the plasma is small solutes. Those would be things like glucose, amino acids, other nutrients that are small and can dissolve in the blood, like gases, ions, and finally waste products. That would be the small solutes. Finally, I'll ask why it's important to know, why do you think it's important to know where the proteins in the blood are coming from? Why would it be important to know that they come from the liver? So I'll just give you a second. So the reason why it's important to know that those proteins are made in the liver is so you can predict what might happen if a person has liver damage. If they have liver damage, then that means that the liver may not be able to make all those proteins at the level that it needs to make them, especially albumin. But also, of course, the transport proteins or clotting proteins. So if you're having a lot of liver damage and you're not able to make those proteins in sufficient amounts, you can have bleeding problems because of that, you can have hormone problems because of that, or you can have blood pressure problems because of that if you don't have the right amount of albumin. And those problems can get very serious or even be fatal. So when you know kind of where things come from, then you can predict what types of damage might prevent those things from being made, and you can kind of predict secondary problems that would arise from different types of organ damage. So we'll look more at erythrocytes, which are the most common formed element in the blood. Erythrocytes have a very large surface area. So most cells are kind of shaped like balls, but erythrocytes are shaped more like discs, and that gives them a very large surface area. Not only are they discs, they're biconcave discs, which means that they have a little depression on each side. Concave means a depression, convex would be the opposite, like a little bulge. So the point of the biconcave shape is to increase the surface area of the cell. When you have a bigger surface area, you can do more gas exchange. Any gases that are going to get in and out of the red blood cells have to cross the membrane. So the more surface area you have, the more membrane you have, the more places you have for things to cross. Erythrocytes are a very unusual cell because they don't have organelles in them, except for the plasma membrane, which technically is counted as an organelle. So they don't have like a nucleus and mitochondria and stuff that all other cells would have, basically all other cells would have. Mostly what they have inside of them is hemoglobin, which you can abbreviate as HB. And the job of hemoglobin is to be transporting gases. Every hemoglobin molecule has 4 subunits in it, so 4 little pieces of protein that are combined to make the whole hemoglobin protein. So here you have little diagrams of the hemoglobin. So each one of these little circles within the hemoglobin would be one subunit. You have 2 types of subunits, alpha subunits and beta subunits. Every hemoglobin has 2 alpha subunits and 2 beta subunits. Then every one of those subunits has what's called a heme group. A heme group is not actually protein, it's a different type of molecule that is attached to the protein subunit, and in the center of the heme group you have an iron atom. And that's where you would actually be having oxygen bind to the hemoglobin. Erythrocytes are also fairly unusual in the body because they don't live very long. You have a lot of cells that don't live a long time actually, but most of your cells do live for quite a long time. Erythrocytes only live for 100 to 120 days, so about 4 months. They're constantly getting damaged as they're traveling through the blood vessels because they're going through under pressure. And some of these vessels are kind of small and they're being forced through there and under a high pressure, and it just kind of bangs them up. There's a lot of wear and tear. And then since they don't have any organelles inside of them, they're not able to repair any damage that they sustain. So normally when a cell gets damaged, it can make some proteins to fix that. But in order to make proteins, you have to have a nucleus, and the erythrocyte doesn't have that. So it can't fix any damage that it sustains. And it just lasts until it has taken too much wear and tear, and then it will die. So I mentioned that hemoglobin's job is to bind gases, and that it binds oxygen at the heme group. Since you have 4 heme groups, you can fit 4 oxygens onto each hemoglobin molecule. And it actually has a different name depending on whether it has oxygen or doesn't. So if your hemoglobin molecule does have oxygen on those heme groups, then it's called oxyhemoglobin. If it does not have oxygen on the heme groups, then it's called deoxyhemoglobin. You can also have carbon dioxide binding to hemoglobin, but carbon dioxide doesn't bind at the heme group. Actually, it binds at the actual protein parts of those subunits. And if you have oxygen attached to the subunits, that's called carbaminohemoglobin. So just like you can have 4 oxygens on every hemoglobin, you can have 4 carbon dioxides, 1 per subunit, but it's just binding at a different part of that molecule. Then finally you have carbon monoxide that can also bind hemoglobin at the heme group, but of course it's not supposed to, that's bad. So carbon monoxide actually binds really strongly to the heme group. It has a very high affinity for that heme group. What that means is just that basically it binds strongly. It grabs it very strong, it doesn't like to let go, it grabs easy and doesn't like to let go. So it's very easy if carbon monoxide is around in the air for it to get picked up by the hemoglobin on those heme groups. And then once it's on there, it's hard to get it off because it binds so strongly. Actually, if you have a hemoglobin where one of the heme groups has carbon monoxide bound to it, and then the other ones have oxygen bound to them, it's actually hard for the oxygen to leave those other heme groups once you have carbon monoxide on one of them. It makes the heme group just grab that oxygen so tight that it won't let go of it, really. So that you end up basically with useless oxygen because it's only useful to you if it will be let go and so it can get to your tissues. So carbon monoxide poisoning is very dangerous for that reason. It basically prevents you from getting oxygen, and if you have like not too much carbon monoxide, then it can do things like make you hallucinate because your brain isn't getting quite enough oxygen. And of course, as the amount of carbon monoxide increases, you eventually will die. So if you ever are having unexplained hallucinations, you might just want to get into the open air. Just go outside for a while and maybe buy a carbon monoxide detector. If you have carbon monoxide on the hemoglobin, that also has a special name, which is carboxyhemoglobin. In this diagram here, you can see the hemoglobin molecule with the 4 subunits. These 2 at the top are the beta subunits, and then on the bottom in the lighter color you have 2 alpha subunits, and each red circle is a heme group, and then the little red ball in the middle is the iron atom. So if you zoom in on that heme group, it looks like this. So this is not a structure made out of amino acids, which means that it's not a protein. It's a different type of organic molecule than with the iron in the very middle, where oxygen or carbon monoxide would be able to bind. So we had talked earlier about how red blood cells or erythrocytes don't live for a long time compared to other types of cells. So because they're always dying, that means that you always need to be making them to replace them. So erythropoiesis is the process of forming red blood cells. And you're always doing this. You're always making actually a very large amount of red blood cells. About 250 billion new red blood cells are formed every day by erythropoiesis. And this would be occurring in your bone marrow, specifically in the red bone marrow. Erythropoiesis is actually part of a bigger process called hematopoiesis. Hematopoiesis is the formation of all the formed elements in the blood. So the red blood cells, the white blood cells, and the platelets. And that all happens in the red bone marrow. So within hematopoiesis, you have three smaller processes. You have erythropoiesis to form the red blood cells, then you have another process to form the white blood cells, and a third process to form the platelets. And it all starts with these cells called HSCs, or hematopoietic stem cells. So in the red bone marrow, you have these hematopoietic stem cells, or HSCs. These are stem cells that have the ability to become any type of formed element in the blood. So it could end up as a red blood cell, a white blood cell, or a platelet. And which one it turns into depends on sort of what line it starts differentiating along. So it has several different options for how the cell can transform into other types of cells that will eventually give rise to red blood cells, white blood cells, or platelets. So in the case of erythropoiesis, this hematopoietic stem cell will transform into a erythrocyte CFU cell, then into a proerythroblast, then into an early erythroblast, then a late erythroblast, then a reticulocyte, and finally a erythrocyte, a mature red blood cell. If it was going to turn into a different formed element, then it would have a different series of intermediate cell types that it goes through. So you do not need to know this whole series of all these cells. You need to know the hematopoietic stem cells where it starts. And then it ends up as the erythrocyte. There is actually one other stage here that we'll talk more about, which is the reticulocyte. So just before a forming red blood cell becomes an actual mature erythrocyte, it's a reticulocyte. The reticulocyte cell is pretty similar to a mature red blood cell, actually. It does not have a nucleus in the previous step. That's when the developing cell loses its nucleus. So the reticulocyte already doesn't have a nucleus. It also gets rid of most of its other organelles. So all these little dots and blobs around here are the organelles leaving this reticulocyte. And it is full of hemoglobin. It has a lot of hemoglobin. Not quite as much as a mature red blood cell, and it doesn't have the shape of the mature red blood cell, but it's very similar. So these reticulocytes can actually enter the blood. Most of them will stay in the red bone marrow and turn into full erythrocytes and then enter the blood, but some of them can also enter the blood just as reticulocytes. Since they have hemoglobin, they can still carry oxygen and be functional, and they'll just mature into red blood cells within the blood, at which point they'll be able to carry even more oxygen. The only organelle that they have left that a mature red blood cell does not have is the ribosome. So they still have some ability to make protein, potentially. The reason why reticulocytes can be important is because they can let you know if a person has recently been making more red blood cells than normal, if they have increased erythropoiesis. So if a person has lost a lot of blood, then that all needs to be replaced. So the process of erythropoiesis will need to speed up. So you're not just replacing the normal loss of red blood cells, but you're replacing like the extra amount that were lost whenever you had that big bleed. So your erythropoiesis is going to go into overdrive, and you're going to be making a lot of reticulocytes. And so you're going to end up having more of them than normal enter the blood. So when you take a blood sample and you count the types of red blood cells that are in that and how much of each type you have, that's called a complete blood count, you'll find that you have a higher than normal amount of reticulocytes. So anytime you see higher than normal reticulocytes on the complete blood count, that's a sign that the person may have lost blood recently. Then the final thing to mention about erythropoiesis is the hormone EPO, which you may remember from AMP1. EPO stands for erythropoietin. It's the hormone that drives erythropoiesis. It's made by the kidney, and then it travels to the red bone marrow, and it tells the hematopoietic stem cells to start differentiating into red blood cells. So it's basically triggering the process of erythropoiesis to occur. So if you don't have the EPO, that means you're not going to have enough erythropoiesis, or you're not going to have any, actually, if you don't have any EPO. So that means that if you have kidney damage, what could happen? You could end up with not enough EPO, and then you could end up with not enough red blood cells, and then you could end up with anemia. So if you have severe kidney damage, it's not uncommon for people in that position to have anemia as well, since they don't have enough EPO. And the final thing to note about EPO is just that it's has been frequently abused by athletes. If you're taking it as a drug, you will be making more red blood cells than normal. You'll have more erythropoiesis than normal. And if you have more red blood cells than normal, then you can carry more oxygen in your blood, and that gives you greater endurance for competitions. So that's one of the things they have to check for in drug tests for athletes. It gives you a big advantage, but there's also side effects. It's not good. So the process of erythropoiesis is closely regulated, mainly by the kidneys. In general, anytime that the oxygen level drops in your blood, the kidneys will detect that, and they're going to be triggered to make erythropoietin, or EPO. Then the EPO is going to go to the bone marrow and tell the HSCs that they need to start differentiating into red blood cells. So you're going to have more erythropoiesis. Erythropoiesis will speed up. Ultimately, you're going to get more red blood cells, and then because you have more red blood cells, you're going to have more oxygen in the blood. The oxygen level of the blood will rise, and the kidneys will detect that it's normal again, and they're going to stop secreting EPO. One more interesting effect of EPO is that it's actually possible for EPO to convert yellow bone marrow back to red bone marrow. If you really need a lot more red blood cells very quickly, then you're going to have a lot of EPO released, and you can actually get some of your red bone marrow back and dramatically increase your ability to make those red blood cells. I guess one final interesting thing about EPO is that it's a hormone that is frequently abused by athletes. If you take EPO injections, then your then your bone marrow will start making more red blood cells. Then if you have more red blood cells in your blood than a normal person has, you're going to be able to carry more oxygen in your blood than a normal person can. And that means your muscles are going to have oxygen to make ATP with for longer than what another person who's not taking EPO would have. So it gives you greater stamina and endurance as an athlete, and it is illegal because it gives you a big advantage in competitions. But there's also side effects. There's bad side effects. Okay, so now we'll talk about the process of erythrocyte death. When your red blood cells are getting old, their membranes get kind of rough and stiff, and it gets hard for them to fit through the smallest blood vessels, which are the capillaries. So they're going to tend to get stuck somewhere in a capillary. Usually they're going to get stuck in a capillary that's in the spleen, and they'll be eaten by macrophages in the spleen. A macrophage is a type of immune system cell. Literally, the name means big eater. And their job is just to eat stuff that could be foreign microbes in your body, or that could be old and dead cells of your body that need to be recycled. So you have a bunch of macrophages in the spleen, and they're going to be eating all the old red blood cells that get caught there. You also have macrophages in, basically everywhere in your body. So if you have erythrocytes that get caught someplace else, not the spleen, then macrophages can eat them there as well. And within the macrophage, you're going to have the hemoglobin of those erythrocytes being broken down. So the hemoglobin is first going to be separated into the heme group and the protein part, which in this diagram is the globin part. The globin, or the protein part of the hemoglobin, is going to be broken down into the amino acids that are making it up, and those will be recycled. The heme group is going to be having the iron removed. The iron can also be recycled, and then you're left with the heme group itself. The heme group is going to be converted to biliverdin and then to bilirubin. Both of those things have colors. Biliverdin is kind of a green color. Bilirubin is kind of a yellow color. And so when you get a bruise, what you're actually looking at is the heme group degradation products, biliverdin and bilirubin. So when you have kind of an earlier stage bruise, it's more green. That's because of the biliverdin. Then as the bruise kind of matures, it'll turn yellow because most of that biliverdin is being converted to bilirubin. Once you have the bilirubin made, it's going to be sent in the blood to the liver. Bilirubin is not soluble in water, which means it's not soluble in the blood. So if it's going to travel in the blood, it has to be attached to something, and it's going to be transported by albumin in this case through the blood to the liver. Once you get to the liver, the bilirubin is going to be modified some more, and it'll end up being excreted in the bile duct into the intestines. And in the intestines, it's going to be modified even some more. It can form a lot of different products, actually. One of those is urobilinogen, and there's plenty of different urobilins. And another one is stercobilin, and there's plenty of different stercobilins as well. So urobilinogen and stercobilin can both be excreted in the feces. Urobilinogen actually has a yellow color, and stercobilin has a brown color. So stercobilin is responsible for the color of your feces in large part. Most of what's excreted here should be the stercobilin. Then that urobilinogen can also be reclaimed from the intestines. It can go back into the portal vein, which is the big vein that leads from the intestines to the liver. So it ends up back at the liver, and it can just go through the cycle again, end up in the intestines again. Or the urobilinogen could leave the liver in the blood and make its way to the kidney. Urobilinogen is actually water soluble, so it doesn't need to be transported by anything. Once it's in the kidney, it'll be excreted into the urine. And since it has a yellow color, that's why your urine has a yellow color. So you don't need to know the names of these degradation products of heme in detail. Just know that their colors are responsible for the colors of a lot of different things in your body, for the feces, for the urine, and for your bruises. Also, if you have jaundice, what you're looking at there is bilirubin. So jaundice is a kind of yellowing of the skin and eyes that's caused by liver dysfunction. So if your liver isn't working properly, then it's not going to be able to modify that bilirubin and send it out in the bile. So you're going to have bilirubin getting backed up in the body. It's not being excreted, and that ends up giving a yellow color to your skin and your eyes. So if you don't have the right amount of red blood cells, that is a problem. Or if something's wrong with the red blood cells so they can't carry oxygen right, then that's also a problem. That's called anemia. So anemia is defined as a decrease in the oxygen carrying capacity of the blood. Usually it means that you don't have enough red blood cells and therefore you can't carry enough oxygen in your blood, but it could also be because of other things. Maybe you do have enough red blood cells, but there's something wrong with them, or they don't have hemoglobin in them, or the hemoglobin that they have isn't the right, it's not formed right, so it can't hold oxygen well. All of those things are going to give you a decreased oxygen carrying capacity of the blood and cause anemia. So if you don't have the right hematocrit, if your hematocrit is significantly below the 44%, that means you have decreased red blood cells in your blood, and that causes anemia. That could happen for a lot of different reasons, usually just because you've been bleeding. If you've bled a lot, of course, that's going to decrease your hematocrit and cause anemia. But it could also be because there's some condition you have that's leading red blood cells to be destroyed. Maybe a cancer or an infection could be destroying red blood cells. If you have a deficiency in certain vitamins like B12 or B9, those vitamins are needed to make nucleotides, and in order to make red blood cells, you need to be able to make nucleotides. So different vitamins that are necessary for, manufacturing basic cellular components. If you don't have enough of that vitamin, you're not going to be able to make enough red blood cells, and you're going to end up with anemia. Also, if you have not enough EPO, you can't make red blood cells without EPO. So if the kidneys have damage and they're not releasing EPO enough, then that's going to cause anemia as well. So this is this is the reason why if you have kidney failure and it's pretty bad, a lot of the time you'll end up with anemia as well, because the kidneys end up not being able to make enough EPO to maintain your red blood cell population. Then apart from not having enough red blood cells, you might not have enough hemoglobin inside of those cells. There's a lot of different reasons why that could happen as well. The most common is that you don't have enough iron. So in general, you're going to be recycling all of the iron, almost all of the iron that is on the dead red blood cells. It's just going to be kept in the body and used to make new hemoglobin for new red blood cells. But there are certain conditions that could lead you to not hang on to your iron effectively, and then if you're not getting enough iron in your diet, you're going to end up with an iron deficiency. Then your body won't be able to make hemoglobin that can actually bind oxygen enough. Then you could have also different problems with your hemoglobin. You could have hemoglobin that just doesn't work right. The most common example of that is sickle cell anemia. In sickle cell anemia, you have a genetic mutation to the gene for one of the subunits of hemoglobin. So that subunit doesn't come out right when it gets made. And that changes the shape of the hemoglobin molecule so that the hemoglobins all form long chains. So in this diagram here, these are chains of hemoglobin that are caused by that mutation. When the hemoglobin makes long chains like this, it's not able to hold oxygen very well, and these chains will also all kind of line up with each other and force the cell to make this strange shape, which is the sickle shape. And then those sickled cells are going to get caught. They're kind of rigid, they have this strange shape to them, they're all pointy, so they're going to get caught in your small capillaries, especially wherever you have like bends and junctions. That's places where they're going to get caught. And when that happens, that causes problems with your blood flow, of course, but since they're pointy-ended, that could also cause pain, so these anemia attacks can be very painful. So we use a test called the complete blood count, or the CBC, to kind of assess anemia and the potential causes for anemia. Also, it's used to assess just different problems with the blood. The complete blood count is literally just counting all the different cells that are in your blood in a sample of your blood and how much of them are there. So you can detect if you don't have enough red blood cells, or if you have not enough of a different type of cell or too much of a different type of cell. So now we'll turn to the leukocytes, or the white blood cells. You have 5 different types of leukocytes in the blood. You can divide them into granulocytes or agranulocytes. The granulocytes are cells that have granules in them. Those granules contain chemicals. The chemicals can do different things depending on what type of cell we're talking about. A lot of those chemicals cause inflammation or kill bacteria or signal to other immune system cells or proteins in the immune system to activate those things. And you have 3 different types of granulocytes in the blood. First you have the neutrophils, then the eosinophils, and then the basophils. So your neutrophils are a very common type of cell in the blood, a type of white blood cell. And here's a standard image of a neutrophil. They have this weird looking nucleus that has different lobes in it, and then those lobes are like connected to each other, so you can see that's all one nucleus. It just has a really weird shape. You can't really see the granules in there too well. They're kind of transparent, but it's filled up with these little granules of chemicals ready to be released. The neutrophil is kind of a general all-purpose immune cell that would respond to any different type of infection that you might have. Then you have the eosinophils. Those are less common. They have these reddish granules or pinkish depending on how the sample is stained. So they look like kind of dark and pink, and then they have this nucleus that also has lobes in it. The eosinophils usually respond if you have a parasitic infestation, but they're also involved in allergies. So people who have allergies have overactive eosinophils. And then lastly, you have the basophils, which are very uncommon. The basophil looks like this. the nucleus is in there, but you can barely even see it. You usually can't even see it because these granules are very dark and they fill the whole cell. So you basically are just looking at granules there. And the basophil is basically going to cause inflammation. So those granules that it has, they cause inflammation, they activate other immune system cells and proteins just to activate the immune response. So actually all of these granulocytes are able to cause inflammation using their granules, but the basophil's like #1 job is just to cause inflammation. All of these granulocytes are also phagocytes, which means that they eat stuff. Usually that means microbes, foreign microbes, or dead cells. The neutrophils are considered professional phagocytes, which means that eating stuff is like their main job. So they also have these granules they can release that cause inflammation or can kill cells, but mostly they're just supposed to be eating cells is their primary function. So next you have the agranulocytes, which are the white blood cells that do not have granules and don't release these chemicals. You have two types of agranulocytes in the blood, lymphocytes and monocytes. Lymphocytes are very special cells in the immune system because they use antigens to recognize specific pathogens. A pathogen is just an agent that causes disease. Usually it's a microorganism that's causing disease. And so lymphocytes are able to recognize specific microbes and attack them. You have two basic types of lymphocytes, B cells and T cells. The B cells are the cells that make antibodies, and the T cells have different jobs because there's different types of T cells actually. Some of those T cells are killing foreign cells directly. They can also kill cells of your own body that have been infected by a microbe and have those microbe cells inside of them. And you also have T cells that serve support roles that are basically just trying to activate other parts of the immune system. Lymphocytes usually have a really big nucleus that fills almost the whole cell and is pretty circular or round in its shape. Then finally you have the monocytes. Monocytes look pretty similar to lymphocytes because they also have a big nucleus that fills most of the cell, but the monocyte nucleus has a divot in it or an indentation. It's not rounded. Some monocytes have a divot that's not that big, so it might be more like just like that, just a really small indent, but they're always going to have something so that it's not just a circular shape in the nucleus. Monocytes, what's special about them is that they can actually leave the blood and enter the tissue, and when they do that, they mature into macrophages. So all of these agranulocytes are phagocytes, but only the monocytes / macrophages are professional phagocytes. So it's actually the macrophage in particular that is a professional phagocyte. The macrophage is like the primary phagocyte in your body. Its main job is just to be eating stuff. Whereas the lymphocytes can eat things as phagocytes, but their main jobs are not that. It's either to be making antibodies as a B cell or to be activating other cells or killing cells as a T cell. Leukopoiesis is the process of making white blood cells. So that's also part of hematopoiesis, and it occurs in the red bone marrow. So it also starts from the HSCs, the hematopoietic stem cells, but in this case, the hematopoietic stem cells are going to start differentiating along a different line to form the white blood cells. And you don't need to know the details of this chart at all, but just that you're starting with the hematopoietic stem cell at the top, and then you're ending with the different types of white blood cells at the bottom. So here you have your granulocytes. Those are made from one line of differentiation, and then your agranulocytes, well, your lymphocytes are made from a different line of differentiation. So one type of cell that I will point out here is the band cell. Those are precursors for granulocytes, and anytime that you have an infection, you're going to end up with extra band cells, because when you have an infection and your immune system is activated, it's going to start responding to those pathogens, to those invading microbes. One of the ways it's going to respond is by signaling the red bone marrow to increase leukopoiesis, so you're making more white blood cells to join the fight. So you're going to have more of these band cells here if you have an infection active. That just shows that the immune system is being activated and a lot of new white blood cells are being made to fight an infection. For your T cells, once they have been made by this leukopoiesis process, they're actually not done yet. They're not mature. They don't mature in the bone marrow. They have to go to the thymus to finish maturing. If they don't do that, then it can be very dangerous actually, because some of those T cells might not be able to identify pathogens effectively. They might not be able to respond to pathogens at all, or they might make a mistake and think that one of your own cells is a pathogen and destroy it. So it's very important that they're able to go to the thymus and finish maturing. Once that process is finished, then all of the T cells that have made it to be actually mature are going to be able to recognize some type of microbe and will not recognize your own cells as being foreign. So all of these white blood cells have different lifespans. Some of them live for not a long time at all, just some days to weeks. Some of them are going to be living for a much longer time. Like your T cells and your B cells are basically supposed to be living for your whole life. So it varies very widely. Next we have thrombocytes, which are important for hemostasis or clotting. Thrombocytes are strange because they're not full cells. They're like little parts of cells. So they don't have a nucleus or anything because it's not a whole cell. They do have some organelles and they have a bunch of granules. So in this image of a thrombocyte, you can see it's just a little packet enclosed by a cell membrane it's from a bigger cell, but it's not a whole cell itself. You have little mitochondria in there giving it energy, and then apart from that, you just have a bunch of these granules that have chemicals that are waiting to be released. Apart from these mitochondria, you're going to have the cytoskeleton, and that's the only organelles that you have in there. So the process of making thrombocytes is called thrombopoiesis. That's the last part of hematopoiesis. It also involves the hematopoietic stem cells, and if they differentiate in this particular way, then they're going to end up as megakaryoplasts, and those megakaryoplasts will transform into megakaryocytes. These megakaryocytes, again, this is all occurring in the red bone marrow, so they're going to be in the red bone marrow, and you're going to have these capillaries passing through the red bone marrow. The megakaryocytes are going to have these little processes or these little arms that they extend into the capillary. And once this arm is extended into the capillary, it's kind of being buffeted by the blood as the blood is moving through. And all the different stuff is kind of hitting those arms. And so you're going to have little bits breaking off of the arms and becoming platelets. So all of these platelets are actually fragments that have been broken off of this larger megakaryocyte cell. So you again don't need to know the details of this, just know megakaryocytes are the cells that are directly forming platelets, and that hematopoietic stem cells are where it all starts. And then that these megakaryocytes are forming platelets by extending their arms into the capillary, and then the platelets get broken off of the arms. The platelets are going to just be circulating in the blood. They're not active unless you have a damage to your blood vessels, and at that point they're going to be activated. They're going to release their granules, and then the clotting process is going to get started. On average, these platelets are going to live for about a week, so you're constantly making them. So here's a summary of the formed elements in the blood. Again, you don't need to know the details of this for the most part as far as the hematopoiesis goes. But notice you're always starting with the hematopoietic stem cell in the red bone marrow, and there's different lines that it can differentiate along. It could differentiate to make the lymphocytes. If it starts differentiating the other way, then it can make your other types of white blood cells, or the platelets, or the erythrocytes. So here you're having, well, I guess this all would be erythropoiesis, then this is thrombopoiesis, and then this is leukopoiesis. Okay, so now we'll get into hemostasis, which is the process of blood clotting, and it's actually, parts of it are kind of complicated. So hemostasis is very important because it maintains blood pressure after your vessels have been damaged. If you didn't have hemostasis, then even if you just got a small little cut, you would actually be able to bleed out from that eventually. You would just continually lose blood until you died. So your ability to live at all is really dependent on having hemostasis that's operating properly. You have 5 basic steps of hemostasis. First, you have vascular spasm, then the platelet plug formation, then coagulation, then clot retraction, and finally thrombolysis. And that's shown in this diagram here. So you start out with your normal blood vessel, and then you have an injury occur and you start losing blood there. And then here you have vascular spasm. In vascular spasm, the vessel is just contracting to get narrower. After that happens, you're going to have platelet plug formation, where the platelets come into this damaged area and are activated and just kind of pile up there and make a plug. Then you're going to have coagulation, which is the part of clotting that we think of as being like clotting. That's the most clotting-y part of clotting. So in the coagulation process, you're forming a proper blood clot. You have all these different clotting factors from the blood coming in, ultimately laying down protein that links these platelets together and makes a really strong clot. So we're going to go through this in detail shortly. You have 2 different pathways by which coagulation can occur. You have the intrinsic pathway and the extrinsic pathway. We're going to go over both of them, and then both of those feed into this right here is the common pathway, which is how it all ends. So by the time you get here, you're actually laying down chains of protein that are binding these platelets together and making a really strong blood clot. So actually, I guess here is where the process has been completed. On all these little yellow lines, those are the protein chains that are binding all these platelets together, also attaching the clot to the vessel walls and making it really strong, holding it in place. Then next, after coagulation, a proper clot is formed. Then you have clot retraction. So in clot retraction, the blood clot is basically shrinking, getting smaller, and that brings the damaged vessel walls closer together. Then your vessel is going to start healing. The walls will be repaired. At the end, once the damage to the vessel is repaired, then you have thrombolysis, which is basically the process of breaking that clot back up. And then you're left with a normal, intact blood vessel again at the end. So we'll start going through these steps. So first you have vascular spasm. Vascular spasm is just vasoconstriction in this damaged blood vessel, which means that muscles in the wall of the blood vessel are contracting. When they contract, the vessel's diameter gets smaller and the vessel constricts, and that's called vasoconstriction. So here in this image, the muscle in the blood vessel walls has contracted, and you can see that the blood vessel is getting narrower because of that. The basic point of this is to reduce blood flow through the vessel so that, like, if you have less blood going through this vessel, you're going to have less blood coming out of the damaged area. Anytime that diameter of the blood vessel goes down, then you're going to have blood pressure go up, but blood flow will go down. Since the vessel is smaller, you can't fit as much blood in there. Because you can't fit as much blood, not as much blood will go through it. So anytime the vessel gets narrower, less blood will go through, and that means you're going to have less blood coming out from this damaged area. Another factor that contributes to vascular spasm is the pressure of the tissue pressing on the blood vessel itself. So as you are losing blood from your damaged vessel, all that blood is ending up in the tissue that surrounds the vessel. That means that that tissue is increasing in its volume and it's going to be pressing onto this vessel, which also further constricts it. Next, we have platelet plug formation. So whenever you have damage to the endothelium, you're going to have this protein called von Willebrand factor released. The endothelium is the simple squamous epithelium that lines the inside of all blood vessels. So whenever these cells are damaged, they're going to start producing the von Willebrand factor, abbreviated VWF, and here it is, the von Willebrand factor entering the blood. That is going to activate platelets that are passing by in the blood. Once these platelets are activated, they're going to accumulate at that damaged area. They're going to bind collagen fibers that are exposed there. So in general, under this endothelium, you're going to have collagen fibers that will also be, of course, damaged by this damage to the blood vessel wall, so that their ends are exposed, and the platelets that are activated are going to come and bind those collagen tendrils and also each other. And the other thing they're going to do once they become activated is release their granules. Those granules end up in the blood, and they can activate more platelets. So as more platelets float by, they're going to be activated by those granules and or by von Willebrand factor, and they're going to come and join the growing platelet plug until you just have a large mass of platelets there that's completely covering the damaged area of the blood vessel. Once you have the platelet plug in place, then you can have coagulation occurring. Coagulation is very complicated. It's the complicated part of hemostasis. The main thing that happens at the very end of coagulation is this protein fibrinogen is going to be activated to fibrin, and it's going to end up binding the platelets in the platelet plug together. So this is the whole coagulation cascade. At the very end, you have this protein fibrinogen, which is one of the clotting factors that is found in the plasma of the blood. It's going to be activated to fibrin, and then it's going to be incorporated into this clot. So you're going to have these threads of fibrin shown in kind of light pink here that are crossing over all of the just all the stuff that's in this platelet. So you mostly are having platelets in here, all the stuff that's in the platelet plug. You mostly have platelets in there, but you're also going to have red blood cells or even white blood cells that get caught up in the platelet plug. All of that is going to get kind of linked together by the fibrin threads, which form kind of a mesh and make this platelet plug into a proper blood clot that is quite strong and hard. So in order to get there, you have to go either by the intrinsic pathway or by the extrinsic pathway. So you can start the coagulation process by either pathway. Then at the end, both of the intrinsic and extrinsic pathways are going to feed into the common pathway. And at the end of the common pathway, you have fibrinogen being activated to fibrin. So for each of these pathways, you have different clotting factors that are necessary. Those would be found circulating in the blood, in the plasma of the blood. Most of them are made by the liver. Some of them need vitamin K in order to be synthesized. So this is kind of the main reason why you need vitamin K in your diet, so that you'll be able to have proper blood clotting. And we will go through each of these pathways in detail right now. Okay, so the first pathway that we'll go over is the intrinsic pathway, also called the contact activation pathway. Contact activation is actually the more appropriate name for it because it's more descriptive of the pathway. Intrinsic is just a shorter word to say. In the contact activation pathway, it all gets started when you have clotting factors in the blood directly contacting collagen fibers that are exposed in the wall of the vessel by the damage that occurred. So specifically, you're going to have clotting factor 12 that is inactivated in the blood, contacting these exposed collagen fibers at the damaged area, and that causes the clotting factor to activate to factor 12A. So for all of these different clotting factors, they're named using Roman numerals, and the little a is attached to the name to indicate that it's activated. So factor 12 means inactivated factor 12. Factor 12A means activated factor 12. A lot of them also have a different name that's not with Roman numerals, but it's actually a word, but you don't need to know those names. We'll just go by the Roman numerals. So, right, in the contact activation pathway, first factor 12 is activated by exposed collagen fibers to factor 12A. Then the next thing that will happen is that your factor 12A activates the next factor in the cascade, which is factor 11. So factor 12A activates factor 11 to factor 11A. Then factor 11A is going to activate factor 9 to factor 9A. Then factor 9A is going to kind of enter this clot and make a complex with calcium. These little green dots would be calcium ions. And it's also going to link up with factor 8A, which is the activated form of factor 8. So factor 8 is actually activated as part of a different pathway that occurs later on. So this whole complex, this whole process is a little bit complicated because of that, or a little bit even extra complicated because of that. But we have some factors that are not even activated or involved yet that will later be activated and activate this factor 8A. So the first time you go through the contact activation pathway, 8A would not be involved because it's not even active yet. But you would have 9A binding to calcium as part of this platelet. Later on, 8A will come in and join this complex. And then the whole result of this complex is going to be to activate factor 10 to factor 10A. So it's actually this factor 9A with the calcium that is able to activate factor 10 to factor 10A. It can do it all by itself, but when you also have this factor 8A in here, it goes faster. So factor 8A is actually a cofactor here, not an enzyme. 9A is an enzyme that activates factor 10, and 8A is a cofactor that helps 9A work faster. So just to go through it one more time again, it all starts with factor 12 activated by collagen fibers, then factor 12 activates factor 11 activates factor 9 associates with calcium and factor 8, and it activates factor 10. Factor 8 makes factor 9 act faster, but factor 9 can activate 10 all by itself. The next pathway is the tissue factor pathway, also called the extrinsic pathway. So again, tissue factor pathway is the more appropriate name there because it's more descriptive, and extrinsic is just faster to say. So in the tissue factor pathway, it all kicks off when you have this blood vessel damage that exposes tissue factor, which is a protein that's found on the membrane of subendothelial cells. So these subendothelial cells are called that because they're part of the subendothelium, which is kind of the layer of tissue that's beneath the endothelium. This is connective tissue down here, and so these are fibroblasts, actually. So these fibroblasts here have actually made the collagen fibers that are all around here in this subendothelial layer, and because of where this tissue is specifically in the body, we call it subendothelium, and these are subendothelial cells. So these subendothelial cells all have this protein called tissue factor that's a part of their membrane, of their cell membrane. Whenever you have damage to the blood vessel wall, the tissue factor ends up being exposed to the blood, and this clotting factor, factor 7, is going to be activated by contact with tissue factor. So it becomes factor 7A. So once you have activated factor 7A, that is going to form a complex with calcium in this platelet, and it's going to activate factor 10 to factor 10A. And that's the extrinsic pathway. So it's a lot shorter than the intrinsic or contact activation pathway. You just have factor 7 activated by tissue factor, then factor 7 complexes with calcium and activates factor 10. As you notice, at the end of the contact activation pathway, we had activated factor 10 to 10A. And here at the end of the tissue factor pathway, we also activated factor 10 to 10A. So they both end in the same place with activated factor 10A. So next we have the common pathway. So at the end of the intrinsic or the extrinsic pathways, we're going to get our factor 10A activated, and we need that to start the common pathway. So both the intrinsic and extrinsic pathways, intrinsic and extrinsic pathways. Both of those are going to feed into the common pathway. So this all is going to start out when you have factor 10A. Factor 10A is going to form a complex inside the platelet plug with factor 5A and with calcium. Factor 5 is actually going to be activated later, so it's another one of those weird ones where this is a cofactor that speeds up the reaction but is not necessary for it to occur. So you have this factor 10A in this complex activating prothrombin to thrombin. Prothrombin is also called factor 2, and then thrombin is also called factor 2A. So this protein here is so important that we're not just going to call it factor 2, we'll also call it by its other name, prothrombin, and then that's activated to thrombin or factor 2A. So it's the factor 10A here that's actually doing this activation. The factor 5A is just speeding it up, acting as a cofactor for 10A. Factor 5A itself is actually activated by thrombin. So once you have had the common pathway run and you've activated thrombin, thrombin is going to activate factor 5 to 5A, and then that's going to come in complex with 10A, and that's going to make this whole process of activating thrombin speed up a lot. So as this process continues, it gets faster and faster. Then the main job of this thrombin is to activate the last factor, factor 1, also called fibrinogen. So factor 1 is also called fibrinogen. That is going to be activated by factor 2A or thrombin to factor 1A or fibrin. Then fibrin is finally this long thready protein that's going to form like a net or a mesh, embedding all of the formed elements in this platelet plug to make it a proper clot. And so we're finally done with coagulation. All right, so what happens in this common pathway, to review it again, so we have factor 2, which is prothrombin, activated to factor 2A, which is thrombin. That's done by factor 10A, and factor 5A helps it go faster. Then you're going to have thrombin, or factor 2, activating fibrinogen, which is factor 1, to fibrin, which is factor 1A. And then fibrin will make threads that bind the clot together, making it a proper mature clot. So here you have another summary of all of that. Over here we have the intrinsic or contact activation pathway. Over here we have the extrinsic or tissue factor pathway. And then at the bottom here we have the common pathway that both of those feed into. Okay, so here at the top of the intrinsic pathway or contact activation pathway, we have factor 12. That is activated by exposed collagen fibers to factor 12A. That then activates factor 11. That then activates factor 9. Factor 9 is going to complex with calcium, also with factor 8, once that is activated, to activate factor 10. Right, so the whole basic purpose here at the end is that factor 9 is activating factor 10. Factor 8 here helps that process happen faster. Factor 8 actually gets activated by thrombin as well, which would be way down here. So by the time you have made some thrombin, you're going to be able to activate factor 8, and then factor 8 is going to help you activate more factor 10, which then helps you activate more thrombin and more fibrin. Then running parallel to the intrinsic pathway, you would have the extrinsic or tissue factor pathway. In that pathway, you have factor 7 being activated by tissue factor to factor 7A. And then this factor 7A is going to activate factor 10 to factor 10A. So both the intrinsic and extrinsic pathways are both going to be happening at the same time. So anytime that you have blood vessel damage, you're going to have exposed collagen. That's going to cause the intrinsic pathway to activate. You're also going to have exposed tissue factor. That's going to cause the extrinsic pathway to activate. So they're both happening at the same time. Why have two? So that the whole thing can go faster. It needs to go pretty fast, actually, so that you don't lose too much blood by the time you've made a blood clot. So once we have our activated factor 10, that is going to form a complex with calcium and with factor 5A once that gets activated, and it's going to activate factor 2, which is prothrombin, to factor 2A, which is thrombin. So again, this factor 5A here is just serving as a cofactor to speed up the rate at which factor 10 activates factor 2. And once you've made thrombin, that is going to then go and activate the factor V and the factor VIII to help the whole thing go faster. So then thrombin is also going to activate your fibrin here from fibrinogens, right? So it takes fibrinogen factor 1 and activates that to fibrin factor 1a. And then that fibrin is going to make the long threads that actually form the clot. Whew. Okay, so now that we're done with the complicated part, the next step in hemostasis is clot retraction. In clot retraction, the clot is basically going to get smaller, and the way that happens is that the cytoskeleton of the platelets contracts. So the cytoskeleton actually has actin and myosin within it. If you remember from AMP1, when you have actin and myosin together in your muscles, they allow your muscles to contract by kind of ratcheting past each other. And it's the same thing that happens in these platelets, where actin and myosin are ratcheting past each other, which makes the cytoskeleton kind of shrink, and then that makes the platelets shrink, and then that makes the whole clot shrink. When the clot shrinks, you're going to have water that was in that clot squeezed out of it, so the clot gets drier. When the clot is drier, that makes it harder and stronger also. And the other effect of clot retraction is that it's going to bring the edges of the damage closer together. So if they start out here and here, they're going to end up maybe here and here. So when the damaged area edges are closer together, that just makes it easier to heal. And at that point, the tissue healing process is gonna kick in, and you're gonna start healing that damage in the vessel wall. After the damage has been healed, then you can do thrombolysis, which is the final step of hemostasis. In thrombolysis, what's basically happening is that the blood clot is being degraded, and you're just getting rid of it. The whole process of getting rid of the blood clot is called thrombolysis, but in order for that to happen, the most important thing that needs to happen is you need to degrade the fibrin. The process of degrading fibrin is called fibrinolysis. So fibrinolysis is the most important part of thrombolysis. Once you've gotten rid of those fibrin threads, then there's nothing really holding that clot together anymore, and it's just going to end up breaking apart in the blood. So this is only going to happen after the damage to the vessel wall is healed. When these endothelial cells are healed, they're going to start releasing this protein called TPA, or tissue plasminogen activator. That TPA is going to activate a protein in the blood called plasminogen to plasmin. And then that plasmin is just going to degrade the fibrin. So it's going to take all those long threads of fibrin and break them up into smaller and smaller pieces until you just have little pieces of fibrin left. So that's fibrinolysis. After that is complete, then there's nothing left holding the clot together, and it's all just going to disintegrate and come apart in the blood. Right, so in fibrinolysis, you have TPA released by the endothelial cells. TPA activates plasminogen to plasmin. Plasmin degrades the fibrin in the clot. That's fibrinolysis. Once that's complete, thrombolysis will proceed, and the clot will dissolve in the blood. Okay, so it's really important to regulate hemostasis closely. You need to make sure that whenever you have damage to your vessels, hemostasis kicks in and it occurs very fast. But it's also important that it doesn't happen when you don't have damage to the blood vessels. If you have clotting happening inappropriately, well, then you end up with blood clots in your vessels, which is very dangerous. So it's really important that you don't have clotting occurring in vessels that are not damaged. So the body has ways of negatively regulating hemostasis to kind of make sure that clots don't form when you don't have vessel damage. So we'll go over some of that negative regulation now. So negative regulation just means that you're stopping the process from occurring, or reducing the rate at which it occurs or the speed at which it occurs. So it's happening less and more slowly. So first you have this molecule prostacyclin. Prostacyclin is going to inhibit the formation of the platelet plug. So you kind of always have prostacyclin around, making sure that platelets don't form a plug inappropriately. Prostacyclin is actually a type of prostaglandin. Whenever you're taking NSAID drugs like ibuprofen, those work by inhibiting the production of prostaglandins. A lot of your prostaglandins are involved in inflammation and the perception of pain, but some of them also are involved in other things, such as prostacyclin, which is involved in making sure that you don't have a platelet plug forming when you're not supposed to have one. So this is the reason why sometimes if you're taking ibuprofen, you can get bleeds as a result, right? One of the main side effects of ibuprofen would be inappropriate bleeding. Okay, so now we'll talk about how hemostasis is regulated some. It's really important that hemostasis is tightly regulated because when you need it quickly, and when you don't need it, you don't need it at all, and you really need it to not happen. So whenever you have damage to your blood vessels, you need to start clotting very quickly. You need to be able to build a big clot quickly so you can stop blood loss. But if you don't have blood vessel damage, then it's really important to not have clotting occurring, because if you do, you're going to end up with blood clots in your vessels that can cause blockage and be very dangerous. So once your clotting has got going, you have a variety of ways to regulate it to make it go quickly. That includes positive feedback loops. So you have 3 examples of positive feedback loops in hemostasis that we kind of talked about. First, you have platelet aggregation, where once a couple of platelets are activated by von Willebrand factor, they're going to release their granules that then attract more platelets. So the more platelets you have activated, the more platelets will be activated in the future. So that's positive feedback that helps you build a platelet plug very quickly. Then in coagulation, you have 2 different positive feedback loops involving factor 8 and factor 5 in the intrinsic and common pathways. So those factors are both cofactors that help some step of the coagulation cascade go faster, but aren't necessary for it to proceed, and they're not activated until the end of the process. So the first time that you're doing coagulation, you don't have factor 5 or factor 8 coming into play because they're not active. Then at the end, you make thrombin, and then thrombin is going to activate the factor 5 and the factor 8, and that makes the whole process speed up and go faster. So that's also a positive feedback loop, where the more coagulation you have done, the more of those factors will be active, the faster coagulation will proceed in the future, in the immediate future, until we're done making this clot. So those are the positive feedback loops that help hemostasis go faster once it's started. But you also have a number of negative feedback or negative regulatory mechanisms that prevent hemostasis from occurring inappropriately. So one of those is prostacyclin, which is a molecule that is made by the endothelium, and its job is to inhibit platelet plug formation. So it basically stops the platelets from being activated and attaching to each other in a platelet plug, and it's always being released by the endothelial cells. Unless, of course, they're damaged, then they stop releasing it. Prostacyclin is actually a type of prostaglandin, which is the chemical that is blocked by NSAIDs. So NSAIDs like ibuprofen and aspirin prevent prostaglandins from being made. A lot of those prostaglandins are involved with pain perception, so when you don't make them, you don't have as much pain. But some of them are involved in other things. Prostacyclin, for instance, is involved in preventing platelet plug formation. So when you're taking these NSAIDs, then you're also blocking the production of prostacyclin, and that's the reason why NSAIDs are associated with greater risk of stroke and adverse cardiac events due to clotting, inappropriate clotting. Then you also have nitric oxide that's also released by the endothelial cells. Nitric oxide is a vasodilator, which means it makes the muscles in the walls of the blood vessels relax, and then the blood vessel will get larger in diameter. So nitric oxide prevents vascular spasm from occurring, and it lets the blood vessels kind of regulate their size locally at a local level without the nervous system being involved. Then you have some other proteins that are all doing different roles well, that all have different roles to play in inhibiting coagulation. So you have antithrombin 3, heparin, and protein C. All of those different proteins prevent one or another step of coagulation from occurring to make sure it happens less quickly. Those would be made by the liver. Right, so all of these mechanisms working together are kind of active all the time to make sure that you don't have clotting that happens inappropriately. Of course, sometimes things can go wrong and you still get blood clots when you shouldn't, which, you know, is very bad. So it's possible to have problems either way with clotting. All clotting disorders involve either too much clotting or not enough clotting. If you have hemophilia, that means not enough clotting. There's different causes for hemophilia, but usually the person doesn't have factor 8 or factor 9, which means that they're not able to complete the contact activation pathway if they don't have factor 9, or if they don't have factor 8, they can still complete it, but they can't do it as quickly. So in order for the blood clot to really get big enough, fast enough to meaningfully stop blood loss, you really need both the pathways. You need both contact activation and tissue factor pathway, and you need the factor 8 and the factor 5 to make the whole thing proceed faster. Alright, so if you're missing any of those factors, then that's going to give you a problem, a clotting deficiency. And in hemophilia, the most common ones to be missing would be factor 8 or factor 9. Then you have thrombosis, which is forming too much clotting. It's forming clots when you shouldn't be forming clots. It can happen in different places, but it usually ends up happening in your leg veins. So clots are just kind of more likely to form wherever blood is moving kind of slow, and it moves pretty slow in the legs. You have a lot of kind of big veins there with slow moving blood, which favors clot formation. So in thrombosis, first you're going to make a clot, an inappropriate clot. That's called a thrombus. So here's this inappropriate clot that you're making, the thrombus. It's getting made at the valve of this vein in the leg. So this valve normally would be preventing blood from going back down, right? Blood needs to go up the legs and the veins, not down, and those valves prevent backflow. But they also make kind of a place where stuff can get caught up. So a lot of the time you'll have clots forming there by a valve. And it's going to stay there. It's blocking blood flow, but not completely. And so you kind of probably don't even know that it's there until the next thing happens, which is a piece of that clot breaks off, which is an embolus. So now this embolus, this broken off piece of the clot, is mobile. It's traveling through the veins, and it's going to go somewhere and get caught. It's going to ultimately get caught somewhere else in the body and block blood flow there, which can cause, that's very serious. So it could go to the lungs. If it goes to the lungs, it's going to cause pulmonary embolism, where part of your lungs don't receive blood anymore, and therefore you don't have enough oxygen in your blood. If it could go to the brain and get caught there, if that happens, then part of your brain will not be getting oxygen, and that tissue may start to die. It could go to your heart, and in that case, part of your heart would not be receiving oxygen, and that's going to cause a heart attack as your heart cells start dying. Right here, you have an embolus that had traveled to the heart and caused a heart attack. And as you can see, it's very large. Right, so those are kind of the main places that a clot might go and really cause serious problems for you. But of course, it could go to other organs. Any place it goes to, you're going to have blockage of blood, lack of oxygen supply to that part of the organ, and tissue death. Okay, so finally we'll go over the blood types, and then we'll be done with this chapter. So blood types are based on antigens on the surface of your blood. Antigens are molecules that can be recognized by the immune system specifically, so by the lymphocytes in the immune system. And every cell has different antigens on its surface. In order to be an antigen, a molecule just has to be complicated enough in its shape that the immune system cells can recognize it specifically and tell the difference between it and a different molecule. So all cells are going to have different antigens on their surface, and that includes your cells too. Different cells have different antigens. Your blood cells have a variety of antigens on them, and they fall into kind of two big groups. So you have the ABO blood group that involves the A and the B antigens, and then you have the RH blood group, which involves the RH antigen, which is also called the D antigen. So all these antigens on the blood cell surfaces are they're oligosaccharides. And an oligosaccharide is just a sugar. Well, it's a type of carbohydrate that's made out of several little sugars. So you can have multiple. You could get a lot actually. You could have more than 5. You could have more like 10 or something, and it can be all different sugars in here. So, well, in this diagram here, I guess the tan sugar is a galactose. The red one is a fructose, and then these green and purple ones are more complicated sugars. But different sugars can make up these oligosaccharides in different patterns, and that is easy for the immune system to recognize. So these oligosaccharides serve as antigens on the red blood cells. In general, you're supposed to make antibodies for antigens that don't belong to you, and you are not supposed to make antibodies for antigens that do belong to you. If you're making antibodies that bind your own antigens, that would be because your immune system recognized your own antigens as being foreign, and those antibodies are going to attack the cells that have those antigens, which would be in this case, your own blood cells. So that would be very, very bad. So an important part of the immune system maturing is just making sure that it's only recognizing antigens that do not belong to you as foreign. So it's important for your immune system to learn that the antigens on your own blood cells are normal and they're of you, and so you don't have a response against them. So in general, you're not going to find antibodies in your own blood that would bind to your own antigens, but you would find antibodies in your blood that bind to other antigens after you've been exposed to those antigens and have had the opportunity to make antibodies in response to them. This is important for blood transfusions because you have to make sure that the person you're getting blood from has antigens on their blood cells that look like yours. If they have different antigens, then your immune system is going to attack those transfused red blood cells, which, can be very bad. So for the ABO blood group, that's the main one we consider when we're thinking about blood transfusions. We also think about the RH blood group, but the ABO is just the most famous one. You have 4 different combinations of antigens that you can have in the ABO blood group. You could be A, B, AB, or O, but there's actually only 2 antigens there, A and B. So this is the A antigen, and this is the B antigen. Everybody actually has the O oligosaccharide also, but this isn't big enough to be recognized by the immune system, so it's not actually an antigen. So you always start out with O here on your red blood cell surface, and then depending on your genetics, either you're going to convert that O to the A antigen, or to the B antigen, or to both the A and the B antigens, or not at all. So if you're blood type A, that means that you have genes that make enzymes that are going to convert all of these O oligosaccharides to A antigens. If you're blood type B, then you're taking the O oligosaccharide and converting it to the B antigen. If you're blood type AB, then you have the enzymes to make antigen A and antigen B, so you're going to have both. If you're blood type O, then you do not have the genes that would give you the enzymes to make either the A or the B antigen, and you end up just staying with the O oligosaccharide that is not big enough or complicated enough to be an antigen. So that means that if you are blood type O, you don't have antigenic blood cells, and, well, at least not for the ABO blood group. So anybody could receive your blood cells and be fine, right? If you're AB, if you're A, or if you're B, blood type O is always going to be safe for you because it doesn't have any antigens on it. If a person who's blood type B gets blood type A blood, the blood type B person is going to have antibodies against the blood type A antigen. And so their immune system is going to attack those type A cells and destroy them. So the person isn't going to get the benefit of that transfusion that they received. Also, if you have a big transfusion, that means that you're going to have like a kind of a massive immune system activation, a massive immune system attack, and that in itself can be very dangerous. It can cause widespread inflammation that actually could lead to shock and death. If you're a type AB person, that means that you can actually receive from anybody. Because you have both possible antigens, A and B, your immune system is not going to attack either of them. So you could get from anybody. Then in the Rh blood group, you either have or don't have the Rh or the D antigen. Right, so sometimes we call it the Rh antigen, sometimes the D antigen. It just, I guess, depends on your preference. If you have it, then you're Rh plus. If you don't have it, then you're Rh minus. And then we combine that with the ABO blood group. So if you're A minus, then you have antigen A and you do not have antigen D. If you're A plus, then you have antigen A and antigen D. So we had briefly talked about what happens if you get a blood transfusion from someone of the wrong blood type. There's also another disease that can occur from incompatibility in blood types, which is hemolytic disease of the newborn. So this occurs in very specific situations where you have a mother who's Rh minus, so she does not have the D antigen, and her child that she's pregnant with is Rh plus, so they do have the D antigen. So since the mother doesn't have the antigen and the infant does, or the fetus does have that antigen, the mother's immune system is going to recognize that antigen as foreign and would respond to it and activate. So the first time that you are pregnant with this Rh positive child, it's going to be okay. What's going to happen is that your immune system is activated, but it doesn't really respond yet because it's only activated just at the very end of childbirth. So actually, normally, you don't have the mother's blood ever mixing with the fetus's blood. So normally you would have this placenta that's kind of making a barrier between the blood of the mother and the fetus. So here you have the mother's blood vessels coming into the placenta, and then here you have the fetus's blood vessels coming into the placenta. And they kind of stay separate from the mother's. The mother's arteries are going to kind of open up into these little pools, and then the blood will be sucked back up kind of into veins and leave the placenta. And then the fetus's blood is going to kind of pick up oxygen and then go back into the fetus. It's not ever directly going to contact the mother's blood, ever. And nothing, it's difficult for things to cross the placenta. So that means that the mother's blood is not exposed to the, is not exposed to the fetus's blood. And so you don't have any interaction here, no activation of the mother's immune system. But during birth, it's pretty traumatic. It causes a lot of tissue damage. So this delicate structure of the placenta is going to get, kind of ripped up, and, a lot of the mother's other tissues will be kind of ripped up, and there's going to be a lot of blood everywhere. And so the blood of the infant and the blood of the mother will be able to mix during that point of childbirth. Also, if some other type of trauma occurs during pregnancy, but normally this would be happening during childbirth. So then you're going to have the baby's blood leaving the baby's blood vessels and entering the mother's tissues. And at that point, the mother's immune system is going to recognize that these blood cells are foreign because they have the D antigen, and it's going to respond to them and make antibodies against them. So that's okay for this baby because this baby is already being born. But it's not good for the next baby if you have another Rh positive pregnancy. Because the second time you have an Rh positive pregnancy, now you have all these antibodies against that D antigen already in your blood. And antibodies are actually able to cross the barrier in the placenta and enter the infant's blood. And they're going to recognize that these blood cells are foreign, and they're going to attack those blood cells and destroy them. And so it's called hemolytic disease because the blood cells are being broken apart. The lytic part means broken apart or destroyed. And so this is very deadly, of course, for the infant because their blood cells are being destroyed. So you can prevent this from happening if you're at risk for it. If you're an RH- mother giving birth or pregnant with your second RH positive child, there's different things that you can do to prevent it from happening. One thing you can do is take steroids. The steroid drugs, corticosteroids, block the immune system. And so that way you can kind of prevent your immune system from responding to the fetus's blood. Or you could inject these antibodies, anti-D antibodies, to kind of coat the fetus's red blood cells so that all the D antigens that they have an antibody attached to it. But these special antibodies that you've injected aren't going to do anything when they attach. They're just kind of sit there and coat the blood cells so that everything is safe. Or you could do plasmapheresis, where you actually remove plasma from the mother's body, filter the antibodies out of it, and put it back in. That's kind of dangerous to do, so you don't do that type of thing lightly. Right, so there's a variety of treatments for this.